Okay, well, let's get started. Today's topic is 3D panfocal microscopy, focus on biomedicine, presented by my colleague, Dr. Gary Greenberg. Gary, go ahead, it's your show.
Thank you so much, Alan. Thank you all for joining us today. I really appreciate it. What you're looking at this beginning screen is just some various shots taken with the system, the panfocal 3D microscopy system. So the agenda for today is I want to tell you what is panfocal microscopy. And then we'll talk about how to publish the results, what the benefits are, some application examples, how to set it up, and talk to you about the optional 3D oblique illuminator, and then we'll take some questions and so forth. So the Edge 3D panfocal microscopy is a plug-and-play, simple accessory that converts your 2D microscope into 3D. It gives you confocal-like results at a fraction of the price of buying a microscope or a confocal-like microscope. The Edge system is compatible with almost virtually any light microscope, inverted microscopes, upright, transmitted, reflected, fluorescence. So it's very easy to adapt to your existing 2D microscope. It's easy to install and simple to use. It consists of a prior scientific focus device and the Edge 3D panfocal software, which goes on to your computer. There's no on-site demo required. You just receive it and hook it up to your microscope. So this is an old 20-year-old Olympus microscope. So what you do is you hook up the camera. So the software controls the camera and the focus device. So the little focus device goes over the focus knob of your microscope and it automates Z-focus stacking. So this is the software screen, and you can see on the left side where the arrow is that that's how you focus the microscope. You can focus using a slider, such as this slider right here, or you can say how many microns you want to move the focus, and you can step it up or down. This can be 10, 20, whatever number you put in here. Or you can use your mouse fine focus wheel, or you can use your keyboard arrows to focus. So there's multiple ways of focusing. As you focus, you will see the position of the focus in the focus position window. So right now we're looking at a Purkinje cell, and we're focused in the middle of it. So what you do is you simply lower the stage to focus to the top of this cell, and you can see we're focused to the top, and you set current position as start. Then you focus to the bottom of the cell. We're focused at the bottom now, and that's 100 microns down. It's a 100 micron section. And then you click set current position as end. Then you tell it what the step size is. The step size is going to be relative to the depth of field of the lens that you happen to use. And then that tells you automatically the number of steps it takes. And then all you do is you push, take a stack. And when you push that, all of a sudden, boom, there's your full Purkinje cell, all in focus, 100 microns from the top to the bottom. So you can clearly see it. That's a 2D projection of a 3D object. So that's why it says at the top, it says that's the view in 2D. You can view it as a 3D model. You can view it in stereo 3D. You can view it in stereo 4D, which I'll show you what that is later. You can do rotational 3D films. You can do anaglyph stereos. Anaglyph is where you wear the red and cyan glasses. Hopefully some of you have some red cyan glasses, which we're going to show you later, some images that will allow you to see in 3D. And you can do a depth map. So basically, you can publish the results in a number of different ways. So most used way of publishing results, is which confocal microscopes usually do, is they publish the reconstruction of the Z-stack. So in this case, that's what we call the 2D view, because that's a 2D reconstruction of a 3D object. And that's how most confocal microscopes will publish their results. Other ways you can publish is, in our system, you can, in one very easy image, you can see and measure the object in 3 dimensions. So it produces a color depth map So you can see blue is in the distance in the foreground, in the background, and red is in the foreground. And the 100 microns from the bottom to the top, and those colors of the dendrites there represent where in 3 dimensions those dendrites are. And the scale on the bottom gives you the XY scale in microns. You can also see it in stereo 3D. If you happen to have your red-green glasses, put the red on the left eye, and all of a sudden you'll see how powerful the human brain is. The human brain turns that image into a very high-resolution 3-dimensional image. Now you can take those glasses off. And another way to see it in 3D, you don't need glasses for this, is you see it as a rotational image. And we create these rotational images after you've made the stack. And this can be either shown at a meeting or at a seminar, or you can present it as a link when you publish papers and so forth. That's a really good way of showing the 3D to your colleagues. So what are the benefits of 3D? Why do we use 3D? The main problem with microscopes is they have a tiny, thin depth of field. This has been for a long time, and that's why confocal microscopes are so popular, because it gets around the problem of a tiny, thin depth of field. So you only see a thin slice that's in focus. So the shallow depth of field produces sampling errors. Think of if the depth of field in a microscope, or when you take sections that are 5 microns thick, or the depth of field is a few microns, think of the sampling error. The diameter of a cell is 10 or 20 microns. So you're not even seeing an entire cell. So this produces sampling errors. So what we allow you to do is look at much thicker specimens, whole mounts, sections that are up to 100 microns or more, and you see it all in sharp focus. So it gets over the depth of field problem. So now you not only see a whole cell in its entirety, but you see the relationships between groups of cells. And this is extremely important in lots of different types of research. So why 3D? Well, confocal 3D imaging has been revolutionizing the biomedical sciences for the last, since the turn of the millennium, actually, for the last 20 years. And the reason is because it improves diagnosis, it increases productivity, and it reveals hidden depth information that you couldn't see otherwise. So who are the customers who need this? Well, hopefully people who are in the biomedical area, neurobiology, pathology, cancer biopsies, developmental biology, 3D tissue culture, anywhere where you're using thick specimens and need to see thick specimens, it's really important to be able to see it all in focus. So let me show you some application examples. One example is to turn your current fluorescence microscope into a full focus 3D microscope. So, for example, this is a triple stain of some muscle cells, and you can turn it into a 3D microscope. So here's an example of a 2D image of a 300 micron thick brain slice. This was using one of these clarity preparations where you can see deeply into the tissue. It's magnified 500 times. Now when you look at it in 3D, all of a sudden we see all the neurites that are there. And by the rotation, you can clearly see the foreground from the background. Here's another example of a fluorescence image. This is a hair root in 2D. This is a DAPI stain, which shows DNA. And there it is in 3D. Now you see all the cell nuclei, and you see in the rotational 3D, you get a good idea of the depth and shape of this root hair. Here's another root hair. This is in hair root. This is in 2D, and now when you see it in 3D, all of a sudden you see all the cells and you see the shape very clearly. Now, if you've got your glasses, please, I'm just going to go through a very quick little slideshow. It won't take long. Put the left eye with the red, and I'll show you some interesting little pictures here. So this is something that nobody else does. It's called Edge 4D imaging. So what this is, it's 3D stereo, and at the same time, it's tilting and rotating. So it's almost like a hologram in a sense. You're getting both the stereo 3D, and which allows you to interrogate the specimen from left and right, but it also allows you to tilt it up and down, so you're interrogating the specimen from different angles. We are unique, and this is a proprietary system. Here's some, this is 135 microns thick, a single neuron from the brain. You can see clearly all the neurites going in different directions. Here's the cortex, a bunch of neurites in the cortex in 3D. There's some small blood vessels and tiny capillaries. And without the 3D, you'd have a really hard time telling what's actually happening there. There's some spirulina in stereo 3D. This is a radiolaria. I'm going to show you a little later how you can get these kind of images using our optional 3D illumination system. That's a diatom. This was evolved a few 100 million years before Buckminster Fuller, who designed these domes that look similar to that. Here's a tiny fungus gnat in stereo. And what it's particularly good at is looking at live cell cultures in 3D. People are doing 3D cell cultures, which is a really popular thing to do right now. It's a really good way to do it. Okay, you can take off your 3D glasses now, and I want to continue to tell you about how you set up the system and then talk about the optional 3D oblique illuminator. So setting up the system is really easy. Essentially, you get a prior scientific focus drive. Focus drive consists of a motor drive, which is on the right-hand side there, and the little control unit on the left-hand side there. And the end of the focus drive has a sleeve, an adapter sleeve, that fits over the focus knob of your microscope. There are a bunch of different sizes that are available. All you do is you tell us which microscope you have, and we send you the appropriate sleeve. You simply take the socket screw and tighten the device over your coarse focus knob, and then the motor focus slides in and attaches to the fine focus knob. So it's really simple to attach. It takes less than a minute, really. So here it is there. You can see that it's not yet attached, and in this next picture you can see that sleeve fits over it, and now it's attached, and now the fine focus, it's not controlling the coarse focus, that's the hold down, so to speak. The fine focus being controlled by that control. And now, once you download the Edge 3D panfocal software, we take control of the camera and the focus to do Z focus stacking. And we take control of the lights. I'll show you that a little bit later. You need a Windows 10 computer. You have to have a fairly good computer. These are the minimum specs for your computer. So I want to tell you a little bit about this optional 3D oblique illuminator. This is a really unique piece of equipment, and it allows you to do things you just can't do with other equipment. It's great for live cell imaging. Prior Scientific have teamed up with Edge to produce these illuminators. They're available. They're just available now. We got our first run, which is almost sold out, and our second run is just starting to be produced now. So this is, again, the same old Olympus microscope. And you can see on the left, all you do is you take the illuminator and you simply attach it just like you would attach the Olympus illuminator. It attaches with one screw. So you just replace your existing illuminator which came with your microscope and replace it with the Edge 3D illuminator. And there's various plugs. You have to plug it into the wall and you plug it into the microscope. Into the software. So once you do that and you're using the illuminator, you'll see that the graphic user interface is a little bit different because we're using the lights. So now it's controlling the lights. If you look over in the left-hand column, you can see it controls the oblique lights. Now, the benefits of this oblique illumination is it enhances contrast, which is why it's great for live cell imaging or anything that isn't stained or doesn't have inherent contrast by staining. It increases resolution of the light microscope, and I'm going to go over that in a little bit of detail in a minute. It improves depth of field, and it produces real-time 3D stereo. Because when you use oblique illumination, it appears as though you're tilting the specimen. For example, if you use oblique illumination from the left, it appears as you tilted this, you're viewing that specimen from the left. if you have oblique illumination from the right, it appears that you're viewing the specimen from the right. So those two images can produce a real-time 3D stereo image. And that can be, you can see on the left side of this GUI, there's a little button that says real-time 3D. And if you push that, it gives you real-time 3D of the specimen you're looking at. So the illuminator, first and foremost, is a powerful contrast enhancement system. So a lot of you may have things like DIC Nemarsky hooked up to your microscope, or phase contrast. Those are powerful contrast enhancement systems, but so is oblique illumination. So take a look at this picture of radiolaria. Radiolaria are not stained, diatoms and so forth are not stained. If you look at it with a bright field microscope, this is what you see. It's not a great image. But if you use oblique illumination with our oblique illuminator, that's that same image. Now you clearly see all the detail of this image with better depth of field, with great detail, let me just look a little bit closer here. Now this is the graphic user interface. So look at the left-hand side where it says lights. So this microscope can be used, or this system can be used just like a regular 2D microscope. If you take the lights on top and it says lights, all the lights, so you see all the lights are on about 80% right now. And so when you slide that slider to the right, it automatically turns all those other sliders, it turns them all up. So this is the picture of that radiolaria up a little closer using all the lights. So that's equivalent to a regular standard bright field microscope. Now look in this left screen. I've turned off all the lights, and now I've turned on only the northwest light. Now with only the north, you can see all the lights are off except the northwest light, the northwest quadrant. And now you get a really good high resolution, good depth of field picture of this radiolaria. In fact, this specimen, as you can see, this was a stacked image. So if you see where it says deep focus mode below the image, you can see that the step size was every 4 microns, that it was 116 microns from the bottom to the top, and there were 30 pictures taken, and they were put together to make that into what we call a 2D view. As I say, it's a 2D projection of a 3D view. Now, if you click something like where it says rotate, that's what you get. So now it makes a little rotational movie loop. So now, not only do you get the great resolution and the great depth of field and all the stacking, but you get a 3D perception of it that you can show to people by posting it on website, on links on the internet. So the 3D illuminator also increases resolution, and it does it by allowing the objective lens to capture higher order diffraction wavelengths. Now I know it sounds sort of strange. How can you get better resolution? That's sort of fixed in the system, isn't it? Well, in fact, it is fixed in the system. This guy Ernst Abbe from Zeiss Microscopes discovered that the limit of the magnification or resolution was reached at the end of the 19th century, and he showed that it's limited by the diffraction of light. And he came up with the first really good formula for D means distance, the smallest distance between 2 dots that you can still resolve as 2 dots and not 1 dot. That's the definition of resolution. Be able to see 2 objects as 2 objects, not 1 object. And you can see the formula has 2 different things in it. It has lambda is the wavelength of light, and it has the numerical aperture of the objective and the condenser lens. Now, I like to show this formula because it highlights the importance of the condenser lens in the resolution of a microscopic image. Most people don't include, most formulas only include the NA of the objective lens, but that's fine if you're just using reflected light, but that's not the case if you're using transmitted light. So the resolution formula, you notice, has wavelength and NA. So let's look at this. Let's look at this picture here of muscle cells. Now you see the muscle cells, they call them striated muscle. That's because they have stripes. And those stripes have a regular distance to them. So if you look at that picture in your microscope, and I encourage you all to do this when you get back to your lab, find a preparation of muscle where you can see the striations. And if you take the eyepiece out of the right-hand side You're looking on the left-hand side, you're seeing the striation. Take the right eyepiece out of the right-hand side, and what you're going to see is the diffraction pattern. You're going to be looking down at the back focal plane, or the back pupil of the objective lens, and you actually literally see the diffraction pattern. And right now you can see that there's a zero order in the middle, and there's a first and second order. The reason the diffraction pattern is lined up in that direction is because it's representing those lines which are lined up in that direction. Now here's what happens: as the lines or the spacings or fine details of an object in a specimen get smaller and smaller, the diffraction wavelets spread out. So here's an example of Pleurosigma angulatum, which is a diatom that is exactly, if you have a really good microscope, you can see these dots in this diatom. They're half micron spacing. This diatom is often used to test how good your microscope is. So in order to resolve that diatom, again, if you see that diatom there and you take the eyepiece out of the right-hand side and you look down the objective lens, that's what you see. You see the zero order in the middle, and then you see the, actually what you're seeing is two six-sided, because there's two diatoms, they're just tilted to one another. One of them is creating six of those, and the other one is creating six of those diffraction patterns. Now, here's something interesting. You notice the blue light is closer into the aperture, so that circle around all the diffraction, that represents the NA. So what happens is, in order, that shows you why blue light gives you better resolution than red light. Red light, some of that red light is actually being slightly cropped out because you're right at the limit of resolution of that light microscope. Now, you can actually see this happen. This is an interesting thing to do. If you set this all up and take the head off of your microscope and put a piece of paper over the back of the objective lens, you'll see the diffraction pattern, but it won't be totally in focus. And then as you raise that paper up, you have to do this in a dark room, you raise the paper up, you will see that that diffraction pattern slowly transforms itself into that image. And a guy named Fourier figured out how to do this mathematically. So the Fourier transforms the diffraction pattern into the image, and the image into the diffraction pattern. So let's take a look at a diatom Amphipleura pellucida. This diatom is just below the limit of resolution, or it's I shouldn't say below, it's actually just at the limit of resolution of light microscope. But you have to play some tricks in order to be able to see it. You have to use oblique lighting. So on top left, you see diatom Amphipleura pellucida taken with a microscope that is not an oil, it's not a 1.4 NA. It's a 0.9 NA microscope, a dry lens. You don't see any markings in the diatom. If you go to a 1.4 NA microscope and you put oil on top of the specimen and oil between the specimen, and it has to be an oil condenser, between the condenser and the specimen, there's oil there also, you will be able to see the lines that are 250 microns apart. So if you really got a good microscope, you'll be able to see those lines. But the problem is the 200 micron spacings, the diffraction pattern, look at the bottom left. For the 200 micron spacings, you get the zero order, but the first order is not being captured by the objective lens, so you can't see it. But if you use oblique illumination, now you tilt the illumination so the zero order is over to the right side of the specimen, and you're getting the first order over to the left side, and all of a sudden, that's what you see. The image on the right there is with oblique illumination, with oil on both the condenser and the objective. And now you can see that those lines were actually made up of dots that were spaced 200 nanometers apart. Now, to show you that actually happens, this is an example of looking at Pleurosigma angulatum, which you can just barely, if you have a good microscope, see the first order. Now, see up on top, we have one oblique light with the zero order down on the bottom right, and you can see there's one diffraction pattern, and it's getting the second order. So now instead of just getting one diffraction order, it's getting two orders of diffraction. And if you use multiple illumination from the right and left, you're actually getting even better resolution because those lights and diffraction patterns are interfering with one another to improve the resolution again. Another thing that oblique illumination does, it improves depth of field. So take a look at this little diagram. If you have a really high NA lens and at A here, and you can see you're fully filling the aperture, you get good resolution, but your depth of field isn't as good. So now if you close down your aperture, you know, you guys, when you go to your to the diaphragm or the iris in your condenser lens, you close down the condenser lens, you start getting a better contrast and better depth of field. But what it's doing, it's making a smaller cone angle. And that smaller cone angle reduces your NA, which gives you not as good resolution, but it gives you better contrast and depth of field. Now, if on the other hand, you use oblique illumination, you are essentially reducing the cone angle of your illuminator, but because you're tilting it, it's getting the higher order diffraction wavelength. So what you have is an increase of resolution, and because the F number is larger, in other words, you created the F number because of the cone size is smaller on one side, you get better depth of field as well. You about double the depth of field. So that makes it really good for things like live cell imaging. So this is an example. This is one of the first pictures I took using oblique illumination. This is looking at the closure of the neural tube. And this is not with stacking. This is just what you get with live. It shows you how good the resolution and depth of field and contrast is with this illuminator. And the great thing is it does this in plastic dishes. Now, a lot of you who look at live cells, you can't use Nemarsky or DIC because the plastic dishes interfere with the polarization filters that exist in DIC optics. So this allows you to see a DIC-like image, but using it with plastic dishes. Here again in plastic dishes, this is just a culture of some cancer cells. Here's a 3D cell culture. I showed you this a little earlier. And again, this is a stacked version, so you're allowed to see it in 3D, but it's using one oblique light from the northeast. So if there's any questions, I think probably, I'm not sure if we have any questions, but probably the first question you all ask is, what is the cost of the system? So I just want to show you very quickly. The system consists of the panfocal software, which is $4,000. You download that. And it consists of the prior scientific Z-Focus drive. That costs $3,400. And if you decide to go with the optional 3D illuminator, that's an extra $6,500. But for those who have attended the webinar today, we're offering you a 10% discount on the panfocal software. So panfocal software will be $3,600 rather than $4,000. So let me see. Are there any? So if there's no questions, I want to just thank you all so much for joining us today. Check out your email boxes. We'll be sending you the email offering you the 10% discount on the panfocal software. If you have any questions later, please talk with either me. You can see my website there, greenberg@edge-3d.com, or EMS can answer your questions as well. So I want to thank you very much again, and have a good day.
Thanks, everybody, for attending today's webinar. If you have any further questions, of course, you can email Gary or myself. Hope to see you next week for the next in the series webinar with the Edge 3D. This one will be on industrial applications or forensics. Have a nice day.